Good afternoon, good morning, and good evening, everyone. Welcome to the session titled Antibody Mediated Prevention Trial, or AMP trial, Where to from here? This is a dual session with some presentations that will start, followed by a roundtable. First up will be Dr. John Mascola from the VRC talking about the history of VRC01, followed by Shri and Naratsu talking about who took part in the AMP trial and where do we go, how did it go for them. So at that point, after those first three talks, I'll come back and introduce the additional speaker. A lot of this data is being seen for the first time, and I look forward to a very robust discussion. Please, Dr. Mascola.
Hey, everybody. My name is John Mascola from the U.S. National Institutes of Health. My appreciation to the chair, Dr. Carl Diefenbach, for the invitation. I'm excited to participate in the Antibody-Mediated Prevention Trial discussion here today. And the organizers asked if I could talk on the derivation of preclinical studies for VRC01. So I'm going to give you a brief overview of how we came to discover and develop the VRC01 antibody. I'll begin by showing a disclosure statement indicating inventorship on an NIH patent for the VRC01 antibody. So I'm going to briefly, in about 10 minutes, overview the isolation of the antibody, preclinical studies, and clinical studies that really led up to the ability to do the AMP study. So this antibody was isolated from a donor that we called donor 45. The work began in 2009. And by screening a number of different HIV-infected individuals from the NIH cohort, we identified this donor who had high neutralization in his serum. And we designed certain probes which allowed us to identify B cells that targeted the CD4 binding site. And then using flow cytometry, isolated those B cells, recovered the antibodies, and expressed those antibodies. And by doing that, we were able to find this particular antibody that we called VRC01. Donor 45 was a slow progressing HIV-infected man who, over the course of 15 years, had viral loads that were in the range of 10 to 15,000 and did not require antiretroviral treatment. Once isolated, we tested the antibody neutralization and found that it was generally quite potent, and we color-coded its potency by this red, green, black kind of phylogenetic tree, showing that across clades, many or most of the viruses was neutralized, and the overall IC50, mean IC50 was about 0.3. Our colleagues, Peter Kwong and Tongqing Zhou, solved the crystal structure, and Julie Wu did the initial isolation, as you see here. I want to just briefly point out, many labs were part of this collaboration. Mark Connor's lab, Rich Wyatt's lab, Gary Nabel, Bill Sheaf, Mario Roeder, and Peter Kwong's lab all participated in the collaborative effort to isolate the antibody. Preclinically, we tested the ability of this antibody to protect in SHIV challenge models, and we did that with two different SHIVs, a SHIV-BAL and SHIV-SF162. And the result of those studies, which I won't go through in detail, were that we found that a serum neutralization titer in the range of 1 to 100, pre-existing the SHIV challenge, were needed to see protection. Later on, after the AMP study was done, further studies in the SHIV challenge model confirmed these data and showed that for a 75% protection overall, a serum ID 50 titers in the range of 200 were what was required on average for protection against SHIV challenge. With regard to clinical studies, we first did a very traditional safety and PK study by studying a smaller number of subjects and dose escalating from 5 to 20 to 40 milligrams per kilogram, as you see here, starting intravenously and then also testing the antibody subcutaneously. This work was all done between the end of 2013 and 2014. And then we worked with the HVTN to do a larger phase 1 study, HVTN 104. And from all of those studies, we came up with the PK curves that look like the ones you see here, showing that over the first month, the concentrations were quite high, above 50, and then as you see a typical decay here, but remaining above 10 micrograms per mil for about 2 months at this dose of 20 mg per kg. And I think you will appreciate that in the AMP study, we ended up using, the design ended up being one group with 10 milligrams per kilogram, somewhat lower than this, and one group with 30 milligrams per kilogram, somewhat higher than this. But the 2-month regimen was based on these type of PK data. We also tested the antibody in HIV-infected individuals. You can see here the normalized viral load graph shows that out of 8 people tested, there was a drop in viral load in 6 of them, so 6 had a response to the antibody. But interestingly, what we found was that the 2 individuals who did not respond had viral envelopes in their plasma, which tended to be resistant in the range of above 10 micrograms per mil. So we had early indications that resistance could be or would be an obstacle towards for antibody therapy. Likewise, in antiretroviral treatment interruption studies with VRC01 alone, there was fairly rapid rebound, indicating that a single antibody was not readily able to suppress the quasi-species once antiretroviral drugs were withdrawn. In order to support the AMP study, which was an experimental medicine study, not a commercial licensure study, we were required to make the antibody, which we had the ability to do at our pilot plant. In total, we filled over 144,000 vials of the antibody and shipped them to all the sites internationally and in the United States that were needed for an interesting number of a total of over 80 kilograms of antibody. I'll just end by mentioning that, of course, this antibody was isolated in 2009, 2010, and then the study was planned subsequent to that. Over the years, of course, there are many, many antibodies and those that are more potent. Just one example of an antibody that's in the clinic is VRC07 that is both more potent, has greater breadth, and includes the half-life extending mutation. So certainly there is room for improvement over and above the results that have been achieved in the AMP study. I want to acknowledge many of my colleagues at the Vaccine Center and many colleagues at NIAID and Division of AIDS who all together planned and executed this study, in addition to my many colleagues in the HIV Vaccine Trials Network and Prevention Networks. Thank you very much.
Hello, my name is Sri Edipaganti and I'm a faculty member at Emory University in Atlanta, Georgia. Today, I'll briefly tell you about the AMP study participants in HVTN 704, HVTN 085. The presentation is about who took part in AMP and how did it go for them. I have no disclosures. The AMP studies are phase 2b proof of concept trials designed to test the efficacy of VRC01 antibody to prevent HIV acquisition. They are 2 harmonized protocols, HVTN 704, HVTN 085, and HVTN 703 and HVTN 081. The randomized placebo-controlled studies were designed to test 2 doses of VRC01, 10 milligrams per kilo and 30 milligrams per kilo against a saline placebo. At week 80, or 2 months after the last infusion, was the efficacy endpoint. At week 104, approximately 2 years into the study, was the safety and tolerability endpoint. HVTN 704 and HVTN 085 study enrolled men and transgender persons who were sexually active with men or transgender individuals living in Brazil, Peru, Switzerland, and the United States. A total of 2,699 participants were enrolled between April 2016 to October 2018. Approximately 900 participants were enrolled in each of the study groups. There were a total of 26 research sites, 19 in the US, 6 in South America, and 1 in Switzerland. One of the most important features of the AMP studies were the early community engagement, along with creation of study websites, social media ads, and animated videos, explaining the concept of antibody-mediated prevention of a viral infection. There was tremendous staff and community enthusiasm since the idea of an antibody preventing infection is at the frontier of new HIV prevention options. There was also an opportunity to make a landmark contribution. In addition, there is no concern for vaccine-induced seropositivity since this was not a vaccine trial. Here are some pictures of community engagement in San Francisco and at Emory University in Atlanta, as well as an AMP flash mob in Lima, Peru. The demographics of the AMP participants are as follows. The gender identification of men in the AMP study, it was 96% men, 5% transgender females, and a smaller distribution of remaining gender identities. A third of the participants were white, 15% were Black or African American, and the other category were mostly from Peru. Half were either Hispanic or Latinx. In terms of the age, half the participants were between 21 and 30 years of age. The baseline sexual risk behaviors in the AMP study, and in particular unprotected receptive anal sex in the 2 months prior to enrollment, were noted in 66% of the participants. 61% were practicing unprotected insertive anal sex. Overall, the median number of sex partners were 3. The baseline sexually transmitted infections in the cohort, syphilis was most common at 13.6%, followed by chlamydia and gonorrhea. Overall, the PrEP uptake was about 40% in the study. Shown on this slide is effective versus detectable PrEP, and it was very similar in the US, Switzerland, and Brazil. Are some pictures of the AMP participants. So how did it go for them? It was overall a very positive experience, as evidenced by a very high retention of 96% for the infusion visits. The annual dropout rate was less than 10%. The study provided access to PrEP. VRC01 was well tolerated. There were beneficial impacts that included personal relationships, medical care, increased knowledge of HIV, and the ability to contribute to medical research. 2% of the participants reported a few negative social impacts which affected the relationships, employment, or insurance, but most were resolved by the end of the study. Just last week, we disseminated the efficacy results of the AMP study to the participants, and here are a couple of quotes from our site in Atlanta. Wow, I'm so pleased to learn how successful this study has been thus far, and I'm excited to see what life-changing medicines will come of it. Very interesting results. I'm glad to provide my little contribution to these important scientific endeavors. And my last slide on acknowledgements. Thank you very much.
Hello, everyone. I have no conflict of interest to declare. AMP, Sub-Saharan Africa, HB10703, HB10081, enrolled women aged 18 to 50 in Sub-Saharan Africa who were at risk for HIV across 20 sites in Botswana, Kenya, Malawi, Mozambique, Tanzania, South Africa, and Zimbabwe. Intense community engagement was utilized to optimize participant recruitment and retention, and we had several strategies that we used. We held stakeholder meetings to provide stakeholders with a platform to raise questions about the study and to give us recommendations about implementation of the study. Here's a slide of AMP in pictures. So between May 2016 and September 2018, we enrolled 1,924 women across Sub-Saharan Africa, and they were randomly assigned to low dose, high dose VRC01 and normal saline placebo, and every woman received infusions every 8 weeks with 10 infusions and 104 weeks of follow-up after the first infusion. So participant characteristics, the demographics show a population at risk for HIV. 97% of participants were Black. Women's ages spanned the full extent of the eligibility age range from 17 to 48 years, with a median age of 26 years, interquartile range of 22 to 30 years, and 43% were under 25 years, and 64% were aged 21 to 30 years. Notable risky sexual behaviors were identified at baseline. Median number of sexual partners in the 60 days preceding enrollment was 2, and reports of inconsistent condom use were high. Almost 17% of participants reported transactional sex at baseline. The prevalence of treatable STIs was high among African women in AMP, with 26% reporting any STI at baseline. The commonest STI was chlamydia, which was reported in 16% of participants, followed by trichomoniasis, then gonorrhea, and syphilis. The prevalence of STIs varied across sites, with Maputo, Cape Town, and Mbeya reporting the highest prevalence rates. The majority of STIs were and remain asymptomatic. So STIs in this population are just but a tip of the iceberg. And we need to remember this as we work on prevention and control of STIs and other comprehensive sexual and reproductive health services for women, including family planning. And in our study, baseline contraceptive use reflects a wide method mix spanning from external condoms, injectables, implants, oral contraceptives, intrauterine devices, and others. Over the course of the trial, 131 women became pregnant, giving us a total of 2,800 person years, or a rate of 5.08%. 12 participants fell pregnant more than once during the trial, and the incidence of pregnancy was highest between women aged 18 and 30. So what was it like for the women who took part, and us, the women who implemented AMP? All I can say is that AMP changed the lives of women in Africa. As you see here, participant 41 from Harare's Milton Park CRS says, "I have come out of the study equipped with great knowledge, and I have discovered who I am." So yes, AMP changed lives in African women. We successfully enrolled and retained HIV uninfected women at risk for HIV. We conducted over 39,000 visits with a retention rate of 95%, and we administered a total over 16,000 infusions with the adherence, an impressive adherence rate of 98%. So this proof of concept study demonstrates that yes, women in Africa can be enrolled in these complex trials. And this proof of concept study really is going to revolutionize HIV prevention, just like what AZT did 20 years ago. I really want to thank the whole village which took part in AMP, especially the women and other participants who took part in AMP. Thank you.
Hello, everybody. It's now my pleasure to introduce Dr. Peter Gilbert, who's the principal investigator of the data center for the vaccine research, the Vaccine Trials Network at the Fred Hutchinson Cancer Research Center. Peter will take us through the study design of the AMP trial. Take it away, Peter.
Yeah, thank you, Carl. And I'd like to begin by acknowledging the many people who contributed to the study. And let's see, I'm trying to advance the slide. There we go. And here I'm especially thanking the laboratory scientists that generated the immunology and virology data, as well as the statistical and computational teams that analyzed the data. Now, when we set out to design AMP, as Dr. Mascola said, we had known from the NHP model that rather than neutralizing antibodies were promising to prevent acquisition of infection, but we didn't have proof in humans. So we set out to generate that proof from AMP, and that would allow us to answer questions about what would the type and level of antibody be required to prevent acquisition, and we'd like to be able to convert information on the type and virus sensitivity of the antibody to a serum neutralization protective titer, which we could then use as a biomarker of protection. This biomarker endpoint could be used for down selection of monoclonal antibody regimens into future trials and for predicting their efficacy. And then also developing a serum titer biomarker would provide a benchmark for vaccine development, giving a sense of what neutralizing antibody titer we need to rank and select vaccines and ultimately to approve vaccines. Okay, and as has been discussed, the schema of the trial with the randomization to 3 arms of lower dose, higher dose versus a control arm in 2 separate cohorts. The primary objective is to assess prevention efficacy of VRC1 versus placebo to block infection through 80 weeks. So this is during the period of follow-up during which infusions are received. And then the secondary objective is to advance toward defining a serum neutralization surrogate endpoint and to get insights into mechanisms of prevention efficacy. So why did the study use infusions? So it was a proof of concept study. So we sought to generate a consistent pattern of concentration of the antibody that would be at a high enough level to provide protection. And we wanted to maximize adherence through the, the participants would come into the clinic for the infusions because we wanted to get an answer on efficacy in this context where there was almost perfect adherence to the product. And then later on, more real world studies could look at effectiveness in the context of variable adherence, say through self-administered antibody in the future. So we used two trials. Firstly, because new biomedical interventions were needed in the two study populations that were selected. And secondly, by studying the antibody in diverse host and virus populations, that's going to optimize the data set for understanding a generalizable surrogate endpoint or correlative protection. So there's really extraordinary diversity in this study across route of transmission, geography, gender, and virus clade. And in using 3 study arms, because the primary analysis looked at the 2 doses of VE-S2R4P pooled versus placebo, we can actually think about it as a kind of a 2-arm trial with VE-S2R4P pooled versus placebo with the same sample size for that, 1 VE-S2R4P versus placebo. But the reason we did 3 doses is 2 doses versus placebo is because it helps us improve our ability to study the neutralization surrogate endpoint by increasing the variability of the antibody concentration and therefore the serum titer that would be occurring against exposing viruses. And when the study was designed, it was designed to have 90% power to detect 60% overall prevention efficacy against all viruses within each trial. And the reason we picked 60 was that, under data that Dr. Mascola showed and that was available at the time, we knew that VE-S2R4 did neutralize between 70 and 90% of circulating strains. And then under a conjecture or that the antibody was providing high enough concentrations that the titer would be high enough to protect at least 60% of viruses, that led to the powering for 60%. Now I'll spend the rest of the time on the secondary objective. And a starting point is data that Dr. Mascola also showed that in the non-human primate model, we were able to document that the serum titer at challenge is clearly a correlative protection. And these non-human primate studies have an advantage for studying correlative protection because you can measure the titer against the challenge virus on the day of challenge. So we can basically prove a correlative protection. Whereas in the AMP studies, we don't see the exposures, so we can't directly look at titer against the challenge virus. And so it's more complicated to try to understand a surrogate endpoint. But essentially, our data analyses were planned to do the best we could in that endeavor. And we divide the types of analyses toward that objective in 3 types. The first we call neutralization correlates analysis, where we're assessing the association of VE-S2R4 serum neutralization against specific viruses with the instantaneous risk of HIV infection. And then secondly, it's neutralization sieve analysis, which is looking at the measured sensitivity of the acquired viruses and asking how does the prevention efficacy depend on that measured sensitivity level. And then thirdly, genotypic sieve analysis, where we're looking at the amino acid sequence features of the envelope viruses and asking how does prevention efficacy depend on those. And the data elements needed to do these analyses, firstly, it's the neutralization resistance testing data and the PacBio sequencing data from the acquired viruses. It's the population PK modeling of the VE-S2R4 concentrations over time for a case control sample of VE-S2R4 recipients. And then we need to estimate the HIV infection dates to be able to impute what the concentration is at infection for each case. And that uses a molecular clock from the deep sequencing data. And then we need to carry that to the end in trying to pinpoint what are the serum titers at the exposures that led to infection against the autologous virus and against other viruses. And then we need to integrate all the data together to get interpretable answers that appropriately quantifies uncertainty. And I won't go through this, but this is kind of the roadmap the statisticians have taken about how we integrate all the data together to try to get answers at the end. And Lynn will be showing some of those answers. And so to say a few more things about how we did this. So the case control data. So the Tamaris lab, her lab ran the Luminex assay to measure the concentration of VRC1 at every single 4-weekly visit for all of the VRC1 breakthrough cases, and then for a random sample of VRC1 recipients who completed 88 weeks of follow-up without acquiring infection. And that constituted the data for doing the PK modeling for the correlates analyses. And this shows what the concentration data look like for the 2 trials at the 2 dose levels. This is for the uninfected VRC1 recipients. So you can see that the concentration stays steady over successive infusions and is similar between the 2 study populations. And we're using a predicted neutralization titer at infection dates instead of measured titer, basically because it's impossible to do the measured titer because we can't get a sample on the day of exposure that leads to acquisition. But fortunately, predicted ID80, which is based on a concentration divided by the virus IC80, is a good predictor of serum titer, and there's more and more data coming out to validate that works well. And to complete the talk here, I'm going to say something about the Sieve analysis. So in the primary paper that's now being published, really the main result is that when we look at the IC80 of all the acquired viruses from the primary endpoint cases, that we can determine that prevention efficacy depends on that IC80. Basically, the VRC1 does prevent acquisition with viruses measured to be sensitive, and then it doesn't prevent infection with viruses measured to be resistant. And then there's a monotone pattern that the more measured resistant the virus is, then the less prevention efficacy there is. And this was featured in Dr. Cour's talk. And we also sought to understand how does prevention efficacy depend on the envelope amino acid sequence. And so the Mullins and the Williamson labs and their teams, they did a huge amount of work in developing PacBio sequencing protocols, and now they're finalizing their data sets on the AMP participants. And we're starting to do the data analyses. And to do the data analysis, the first thing we wanted to do is define what we call a proteomic antibody resistance score, which is a score for an envelope amino acid sequence that predicts whether their virus is resistant to VRC1 based on the TZMBL target cell assay. And so we did that, and we got a highly predictive model. And then what we did is we repeated the neutralization Sieve analysis, except using this proteomic antibody resistance score. So I'm showing results here where this black curve is the predicted probability that the virus is resistant, based on the sequence of the virus. And you can see that the efficacy starts at 80% against the most sensitive viruses, and then goes down to 0. And interestingly, the result mirrors what we got with the measured IC80 marker. So yeah, basically we're concluding here to say that we sought to do this study to get proof of concept whether a broadly neutralizing antibody could prevent HIV infection, and then to see if we could define that prevention efficacy by the serum neutralization level, the virus resistance level, and the virus sequence features. And I think there has been progress toward those objectives to date. Thank you.
Thank you, Peter. It's now my pleasure to introduce Dr. Lynn Morris, who will speak to us about the relationship between ID90, ID50, and acquisition. Peter alluded to some of that data in his talk that was just concluded. And then the final speaker of this part of the session will be Dr. Larry Cour, who will talk to us about what does this mean for antibody-mediated prevention for the future. So, Lynn.
Thank you very much for the invitation to give a presentation on the AMP trial and some of the laboratory and statistical work that we've been doing on this trial. So the AMP trial was designed to assess whether VRC01 could prevent HIV infection. And as you will have heard last week from Larry Cour, while there was no overall prevention efficacy in this trial, there was efficacy against the most sensitive viruses. And so that's where the laboratory objectives become really important, because these were to determine how viral sensitivity to VRCA1 impacts prevention efficacy, and also to see whether we can establish serum levels of VRCA1 that are required for prevention efficacy. Just to say that this trial has just recently become unblinded, and so there's lots of other additional efforts that are ongoing. So just to tell you about the laboratories involved in the study, the Africa trial, the sequencing and founder variant selection was done in Carolyn Williamson's lab in Cape Town, and the neutralization was done in my lab in Johannesburg. The Americas trial was done by Jim Mullins and David Montefiore. Just to say, there was lots of oversight as well as QA, and also lots of interaction with the statisticians at SHARP. So we assessed the VRCA1 sensitivity as the infections were accruing, and so samples were sent to the sequencing labs, and they identified the founder sequence. We synthesized the gene and generated the pseudoviruses, which we tested against VRCA1 in the TZMBL neutralization assay. And the hypothesis that we were working on is that in the VRCA1 arm, any breakthrough infections would be resistant to VRCA1, whereas in the placebo arm, they would mostly be sensitive viruses. So in total, there were 175 endpoints in this trial. We were able to do the in vitro sensitivity in 93% of them. And of those, 90 were from the Americas trial and 72 were from the Africa trial. And just to say, the results are going to be combined from now on for the rest of the presentation, because we didn't notice any big differences between the trials. So overall, 40% of the infections were in the placebo arm, 33% in the 10 milligram per kilogram arm, and 27% in the 30 milligram per kilogram arm. So the viruses were also categorized based on their sensitivity, and this was predefined categories. So the most sensitive viruses are in cyan, and as you can see, the 30% of the viruses in the control arm were sensitive to VRCA1. So that's 19 viruses. There were 4 viruses in the 10 milligram per kilogram arm and 5 viruses in the 30 milligram arm. So fewer viruses in the VRCA1 arm that were sensitive to VRCA1. And as you can also see that there was more resistant viruses in both the VRCA1 arms. So these data were used to calculate the prevention efficacy. So you can see that the 19 viruses in the control arm and 9 in the VRCA1 arms gave an overall prevention efficacy of 75.4%. And that lasted for the 80 weeks of the trial, as you can see by the cyan line on the graph. There was no prevention efficacy in the intermediate or the resistant viruses, and again you can see that on the graph on the right. So another way of looking at this is to look at the individual IC80 values of each virus. And as you can see in the control arm, there were a large number of viruses that had a sensitivity of less than 1 microgram per mil, and far fewer in the VRCA1 arms. And these would be the viruses that would have been blocked by VRCA1. But overall, you can see that the median IC80s of the viruses in the VRCA1 arms are higher. In fact, they were 2.4-fold higher than viruses from the control arm or the placebo arm, again indicating that there was more resistance in the VRCA arms. So in summary, prevention efficacy against sensitive viruses was 75%. there was no overall PE in this trial, and that's likely because only 30% of circulating viruses are sensitive at that concentration. We didn't see a difference between the two trials, so suggesting there was no impact of gender, population, clade, dose, or route of acquisition. But of course, the number of sensitive viruses, is very low to make that assessment. And then very importantly, the TZMBL neutralization assay could predict the in vivo efficacy. And that's really important for going forward in terms of thinking about selecting the next BNAbs, that we can rely on the in vitro assay to help to guide us. So the next thing we wanted to do was to see if we could define the serum neutralization titer that can predict the prevention efficacy. So as I've just shown you, we know the IC80 of all the VRCO1 breakthrough viruses. We can calculate the relationship between the VRCO1 titers using ELISA or BARMA and the neutralization titer, the ID50 and the ID80s. And using these, we can predict the VRCO1 concentration at the time of infection, and this is done using PK and HIV diagnostics. But just to say that this is a particularly tricky thing to do, particularly around the time of the infusions, and also the timing of infection is very difficult to do. So there's lots of ongoing validation work there. Nevertheless, using all of these parameters, we can estimate the serum neutralization titer that's required to be effective against viruses. So I just want to tell you a little bit about that. This is the pilot study that we did together with Yunda and Peter. And to say the ELISA data is published and the neutralization data is in press. And I'm going to just focus on the neutralization data here. So we selected a small panel of viruses from the placebo arm of the trial, 2 subtype Bs and 2 subtype Cs. And as you can see, we chose 2 intermediate and 2 sensitive viruses. We also included a reference virus and then a very sensitive virus as a positive control. And then we measured the serum neutralization titers against this panel in 31 uninfected recipients at 8 time points, so over the course of their infusions. And this just gives you a sense of the actual data. So this is the blue is the Africa trial and the red is the Americas trial. And as you can see, these typical sawtooth profiles of the levels of VRCO1 over time. And you can see that the titers are higher in the 30 milligram arm. And you can also see that the titers are higher against the more sensitive viruses. And in fact, it was very hard to measure anything in the 10 milligram per kilogram arms against the more resistant viruses. So we next wanted to use these parameters to look at the distribution of neutralization titers to the transmitting viruses at the time of infection in the infected and the non-infected groups. So as you can see, the titers in the infected participants, the median titers are lower compared to the non-infected participants, and they're also lower in the 10 milligram per kilogram arm. So basically showing that in the VRCO1 infected cases, there was lower serum titers to the transmitting viruses. We then also looked at the serum neutralization titer and its association with prevention efficacy at week 80. So as you can see on the y-axis, the prevention efficacy, and the x-axis is the neutralization titer. You can see increasing prevention efficacy with increasing VRCO1 titers. And then focusing on the 75% prevention efficacy that we saw against the sensitive viruses, you can see that the titer to get 75% PE is around about 200. I mean, there's obviously a range there, but that's very similar to the titer that was seen in the meta-analysis of the non-human primates, which was for 75% prevention efficacy was 219. So just in conclusion, AMP has provided proof of concept that a monoclonal antibody can prevent HIV acquisition. Both viral sensitivity and serum neutralization titers were strong predictors of prevention efficacy. And the predicted titers needed for prevention efficacy are in a similar range to the NHP studies. So finally, I'd like to thank everybody that made this study possible. It's really been an incredible study to be involved in. And first and foremost, to thank the AMP study trial participants who are incredibly diligent in coming regularly to all of their clinic visits, but also the community advisory boards and the community members. And to thank the huge teams from both trials. And I think you will see the long list in the next talk of all those people involved in the trials, and of course also to thank our funders, but to really also thank Larry and Mike for the incredible vision and leadership that they've shown in getting this trial to completion. And then specifically for this talk, I'd like to thank the statisticians, Peter Gilbert, Alan de Camp, Yunda Huang, Michael Juraska, Paul Edelfson, Elena Georgi, and then also to thank the lab PIs, or the main lab people, David Montefiore, Nonom Kize, Carolyn Williamson, Jim Mullins, Georgia Tamaris, and John Hurrell. And with that, I thank you for your attention.
In this last talk of the symposium, I'm supposed to review the big picture. What do these data mean for the future of antibody-mediated prevention? I thought for the future we'd look at a target product profile for a monoclonal antibody cocktail. A high prevention efficacy, 90%, no toxicity, administration frequency that is ultra long, 4 to 6 months, 6 months ideally, and a durability with no functional anti-drug antibody of over 3 years. The AMP trial gives us 2 in vitro guideposts to get to this potential target. The IC80 of the isolate, less than 1 microgram, we've gone through that. And the ID80 in the serum, the amount of neutralization in the serum that we need to achieve to ensure high efficacy. The IC80 of the circulating isolates been gone through many times, and I'm not going to dwell on it. These are the data that have been shown multiple times during both this symposium and in prior symposiums of less than 1 microgram with a 75% efficacy. This curve shown earlier in the symposium reviews the ID80 at the median midpoint concentration. So essentially, what's the ID80 in serum that we would want if, let's say, we're giving it subcutaneously or intramuscularly, that would achieve a high prevention efficacy? In this 80 to 90%, that comes out to be 130 or 140 as the infectious dose 80 that is of serum that would inhibit isolates that would give you essentially 95% effectiveness. Now I'm talking about the ID80 in serum as intriguing because it gives us an absolute number that would inhibit circulating isolates with just 1 antibody. Now, if this benchmark is achieved by some cocktail in which both antibodies cover the same isolate, then the relative contribution of each antibody may be reduced. If it requires a cocktail which, because of its breadth to get to the IC80 over the entire population of circulating viruses is such, then each antibody would have to essentially achieve this serum level of ID80 of 150. And the second I'm dwelling upon it is that the non-human primate data suggests that it is the ID80 value that is the mechanistic benchmark, irrespective of the site of neutralization. And that has been published previously in this meta-analysis. This is a slide from that of these 265 non-human primates receiving a single BNAP from 12 different non-human primate studies. And what it shows here is that irrespective of the site of infection of the BNAP, it was essentially the serum, this 50% neutralization titer, the ID50, that is associated with protection. It was about 75% protection at an ID50 of 130. So providing the sort of the mechanistic issue wasn't the site of action. It was what's the concentration of antibody that was effective in this essentially high dose mucosal challenge series of animal models. Now, it is possible this marker may be too high with the LS antibodies, which concentrate in tissue more than the non-LS antibodies, and which block in vivo infection, which we think is better than that. So it is possible that we would overshoot that, but maybe that's what we actually want to achieve in the initial trial. Illustration of this are these beautiful tissue biopsies staining for VRC01 in the top and VRC01LS in the bottom from HVTN116, a study that Julie McElrath and her colleagues conducted, which took biopsies from people after the first dose of these two respective antibodies. You can clearly see way more antibody in the bottom panel with VRC01LS and extending for a longer period of time. This is in rectal tissue. In vaginal tissue was the same. The ex vivo challenged these with sensitive strains and somewhat less sensitive strains of virus. And here's a Kaplan-Meier curve that's shown that inhibition of challenge or relative protection is much longer at the high dose VRC01LS as compared to even the low dose LS, as well as the high dose VRC01 per se. People who did this, especially the lab work, Rina Astronoma and Mario Lemus and Julie's group, deserve credit for a marvelous study, as well as the clinical trial sites that did this work, both in Cape Town and now getting to this ID80, we can do this through a mixture of 3 antibodies. There's a single trivalent, or it could be by various different bivalent preparations. This slide, I think, has been shown earlier in the symposium that shows the coverage essentially at less than 1 microgram from VRC07 529LS, the lead there. The coverage would be close to 80% of isolates, and then PGT121 and PGTM1400 that have complementary isolates as to what they cover at approximately 50% at less than 1 microgram. Using those 3 antibodies, you're well over 94% in the coverage from the circulating strain, and actually 70% of those isolates would have double coverage. This is just to say that the trispecific antibody has been published. It's been in clinical trials. It appears to have in vivo activity in HIV positive persons. It's being modified, and second generation is being made, and hopefully clinical development of the single antibody with 3 FABs will be in the clinic relatively soon. This is a depiction of the bispecific antibody that David Ho is developing. The method of administration also needs to be worked out. How much antibody is required to achieve this level of ID80? What's most cost effective, subcutaneous versus IM versus IV? There's a tension between the bioavailability versus the administration costs. If subcutaneous is only 30% bioavailable, then one needs large volumes also and 3 times more drugs regarding the cost of goods. Can we concentrate the antibody so it's enough to deliver IM? And can we give it IV push or a short infusion time? There are infusion machines that have been developed from low and middle income countries, and all of this needs to be worked out. Lastly, I would end by saying the AMP data gives us momentum. COVID-19 has shown us that when there's a will, there's a way. When we work together and want success in a truly public health and collective way, we've taken vaccines and monoclonal antibodies from discovery to EUA in an 11-month period of time. We in the HIV field need to pull together to drive these programs as expeditiously as possible and to see if this technology can make a difference in HIV prevention. Lastly, I want to make sure that we continue to congratulate the protocol teams, the chairs, the co-chairs, the laboratories that have done incredible work in all this in these monumental studies. HPTN 704 and the 703 team, and the participants who participated in the trial. Thank you.
I would like to thank all the speakers that we've had, and now we're going to be add to our panel with 3 additional individuals to round out this part of the session. First up will be Georgia Tamaris from Duke University, Michelle Nusenswag from Rockefeller University, and Myron Cohen from University of North Carolina. I believe you're supposed to give some short remarks, and then we'll get on to some Q&A. But before we do that, I just want to reflect on what Larry just said about through the AMP study and the work that the VRC did in making the antibodies really proved that antibodies could be used, at least from a safety perspective, quick and could move quickly. And this really set the stage for what we were able to do with the monoclonal antibodies that came from Regeneron, from Lilly, and from other companies that are currently either at the EUA stage or in trial. So I think we have made some significant contributions here, and now we need to discuss the way forward in this panel on HIV. So Georgia, do you want to make a few comments?
Sure, thank you, Carl. So again, congratulations to the team on the remarkable study and for providing really clear answers for the field. I think it highlights going forward as a proof of concept for vaccine strategies, in particular for broadly neutralizing antibody vaccine strategies, the need to develop programs that are eliciting antibody responses against multiple epitopes, including for broadly neutralizing antibodies, multiple specificities. Also the consideration of something we've talked about in this trial, in combination trials for BNAbs going forward, as well as for vaccines, the role of antibody Fc effector functions and how those may have helped in this trial, or how they may be leveraged in future combination BNAb studies that optimize the Fc function for targeting more viruses in this case than those that were most exquisitely sensitive to the broadly neutralizing antibodies. So I think we're learning from that about how those may be applied to future studies for fully engaging all the antiviral functions of antibody responses.
Thanks, Georgia. Michelle, do you want to make a few comments?
Yeah, I do think that the team that did this has to be congratulated for being able to achieve this under, what had to be very challenging conditions. But I do think that the results of this trial are disappointing. I think we should all acknowledge that. I think the disappointment comes from having made an initial prediction based on pseudovirus tests, which are inaccurate in predicting the activity of the antibodies in vivo. It's also clear from the other studies that were done in people, the ATI studies, that VRC01 had very little or nearly not measurable activity in preventing viral rebound. And that is entirely consistent. That result was entirely consistent with what happened in AMP. At the same time, there are other antibodies that have been shown to be effective in vivo. And so I think one has to be disappointed about these results, learn what we can, but I would say that it would be difficult to say that AMP will really inform other antibodies based on the pseudovirus tests. And the issue about vaccine is also difficult since this is just a single monoclonal antibody.
Thank you, Michelle. Mike, do you want to make a few comments?
Yeah, thanks, Carl. Just very briefly. Well, first, I again congratulate all the investigators who worked on all this, and thanks to the NIH and others who helped support the study. I just think there's 3 things that I would say about this. First, I think it's tough not to be enamored of this incredible science over the last 10 years. I think most of us have been really admired Michelle's work and John Mascolo's work, and ignoring any conflicts about the details of how the work is done. It's just really compelling science, and I think that's exciting and gets people excited about being scientists. I think the AMP trial, well, I'll disagree with Michelle. We needed to know how to do this. You know, there was at the time when we started the chance of leaving the train station or waiting 4 more years to stay in a train station. We left the train station, and the degree of success that was achieved is not just about making a product, it's about understanding how we would do this or could do it in the future. And as Carl indicated, it truly informed everything we've done in the COVID space. Lastly, I have a different responsibility about a bigger picture. You know, we have to look at the monoclonal antibodies in the context of other agents. And the other agents are oral agents that are very long acting. Islatravir, a pill that lasts a month and maybe an implant. Cabotegravir, which the NIH helped to develop, which is a 2-month injectable agent. Lenacapavir, a drug we're starting to work on in collaboration, we hope to work on in collaboration with Gilead. So I think that as we think about monoclonals going forward, we have the science part, and then we have the kind of reality-based, well, what are we going to do to stop the HIV pandemic in developing these agents? So I think that's it's a good place to be, to have so many choices at this point. So I'll return it back over to you, Carl.
Well, thank you for that. Let's, because this is now a panel of the speakers as well, let's start with maybe some discussion about from some of the other speakers about if they have any reactions to what they've heard and if they have anything they would like to say. So what I need to do is see if anybody wants to raise their hand and speak. Okay.
Thank you very much, Carl, and thank you to the organizers for giving AMP this opportunity, the AMP team this opportunity to present. I really would like to reiterate what Mike has just said, that we are in a very good place. We did this study exceptionally well. If I recall, maybe about in 2016, when I really made my first presentation representing Sub-Saharan Africa, I, as the protocol co-chair, was anxious about, would we be able to do this in Africa? Would women come? They are busy women, you know, they are doing work and they're looking after families. Would they come and sit and have infusions every 8 weeks and come to clinic every 4 weeks? So I didn't think it would be this successful. So some might view the results of AMP as being disappointing, but as the protocol team, we are really, really excited that this is a proof of concept study, and we've proven that, yes, monoclonal antibodies do work, and so we are in a better place. So I just thought I would start by really thanking our participants, our communities, all the stakeholders, and the entire village that took part in AMP. Thank you. Over.
Thank you, Naratsu. So let's come back to this issue of how we can use the results from the AMP trial and whether they can be used to predict what we need from a cocktail in the future. And spend some time talking about the target product profile that Larry showed. Can we think about what we saw from AMP and take measures from other antibodies and talk about what can we expect from a mixture of 2 or 3 additional of other antibodies? Just one other thing to point out is that when John spoke, he clearly showed that we had VRC01 was a first generation CD4 binding site antibody, and the level of improvement has been nothing short of spectacular. And the other forms are in more advanced clinical development as well. So I'll stop there and open this up. If somebody wants to raise their hand and make a comment. Go ahead, Larry.
You know, no one is saying that the pseudovirus assay measures everything, but a biomarker is a biomarker, how much mechanistic it is or not. I think the graph that Lynn showed, which is the non-human primate collation of 252 non-human primates with 12 different studies in which we essentially got the same curve, irrespective of what antibody was given in the challenge is a very helpful sort of corollary, that when you see the same kind of slope curve out of the non-human primate, and we see this also in the first human study, that it sort of corroborates the basic issue here, that there is some predictive value from the assay. You know, maybe Peter wants to go at that. That doesn't mean there aren't other assays that, Michelle, that won't have predictive value, might even have tighter predictive value, might have a slightly different cutoff point. But we do have enough of a calibration to give us some confidence that, you know, we should de-risk this. In the end, no one is saying you don't do a clinical trial. I myself looked at this and sort of said, and I raised my own talk, that, you know, tissue concentrations probably do make a difference, and the LS mutations clearly give better tissue concentrations and longer tissue concentrations, and maybe the serum cutoff won't be what it is. Maybe the in vitro cutoff actually will be what it is. Proof of the pudding will be in continuing to explore. And I think Mike made the point that the technologies allow us to explore, and these are technologies that are expandable and can be translated in an accurate sense to be correlates in large-scale clinical trials.
So a question to you, Lynn. Would you want to expand at all on what Larry just said about the curves for non-human primates, and maybe flesh that thought out a little bit more?
Well, actually, I wanted to comment on the the assay.
Okay.
So you know, the pseudovirus assay is more sensitive than the PBMC assay. We would have missed the signal, I think, if we had used PBMC grown virus. But just to say, Michelle, that you know, I mean, every assay is artificial at some level, and you know, that the PBMC grown virus, you know, will also have some culture changes. And also, I think you also include a cell line in your culture. So there is probably some virus growing in cell lines there as well. But just to maybe reassure you, we are going, we actually can't do the PBMC grown virus assay in AMP because we don't have PBMCs from the AMP trial. But we are going to make infectious envelope clones and you know, and try and simulate that to look at that. But I guess the reason for the TZMBL assay is simply you know, the standardization. I mean, it's really revolutionized the field and the things that we can do. I mean, it's just been a remarkable assay. And I don't think we'd be this far along if we didn't have the TZMBL assay. So I do take your point, and we will try the, you know, as I say, the infectious molecular clones, because that's the only option that we can do in this trial.
Yeah, I don't, Lynn, I don't want to say that TZMBL assay is no good. It's an outstanding assay, as you just pointed out, and it really has moved the field forward. It's just in making predictions about what's going to happen clinically that assay, you have to take into account the fact that it is far more sensitive than and that it varies, the sensitivity difference varies with antibody. So for example, PGDM 1400, which Larry mentioned as part of a triple combination, is 100 times less active on real viruses than it is on pseudoviruses. Its brother, the 256, CAP 256, there's a far smaller difference. So if you are going to predict based on pseudoviruses and use PGDM 1400, you're going to make a huge mistake. If you use the pseudovirus assay to predict for CAP 256, you may not make such a mistake. So just want to say that in moving forward, this is really for the future, that more than one sort of assay should be used on circulating strains in order to try to understand how the antibodies will perform.
There are a lot of questions in the chat. about next steps, future designs, all kinds of activities about what would the next antibody trial look at. But before we get to those questions, and I'll make sure we spend enough time, leave enough time for that, I have a question for the group, that there appeared to be some modicum of protection early in the trial against moderately resistant viruses, and then it seemed to wane. What lessons can we take from that data, and what questions does that raise about the future of either antibodies, or what does it tell us biologically about what's happening in vivo? So it's a speculative question. Peter.
Yeah, so I can get started on this question about, the data did show that it looked like against the viruses that were of intermediate resistance level, that the prevention efficacy started out at 50% or got there quickly, or 50, 60%, and then waned away to zero but by the end of the trial, and what could explain that? So a potential explanation would be that the concentration of the antibody weakened over successive infusions, and we were able to rule that out based on the PK analysis. Another possibility would be loss of functionality of the antibody, of neutralization over time, of successive infusions. And we were able to show that that wasn't the case for neutralization based on TZMBL assay. And then there's another potential explanation of ADA, anti-drug antibody, which could emerge during the later part of the trial. And right now that's being tested, so we don't have the answer yet. Also in these FC trials, there's a possible spurious waning based on a depletion of susceptibles that happens more rapidly in the placebo arm. That can occur if there's a high risk of infection in subgroups, like at KwaZulu-Natal, for example. And then basically the most at-risk people get infected first, and then you can end up with a bias about FC over time. It's going to be really hard to document whether that explanation could explain it, because to really explain it, we would need more data on risk behavior from the participants than I think we have. There's also a possibility that that the waning that we're seeing is it's in terms of detectability of infection, but we actually are not seeing waning in terms of acquisition of any infection that we weren't able to detect, because we do have some evidence that there could have been mass infections. We don't know if this is occurring, but what I mean by that is that for individuals on the higher dose of VRC01, it's possible that some of them had their HIV diagnostics register negative, even though they're infected, because the virus, the antibody was keeping the virus suppressed during the continual infusions. And then later on, when they discontinued infusions, when the antibody was gone, then the virus emerged and it became detectable. And we do have some hints that could be true based on we do have data that in the higher dose VRC1 group, there was basically more cases that occurred in the tail of the antibody at the end of the study after discontinuation of the antibody than in the placebo arm. So, and then, yeah, I'll stop there. So that's some of the thinking that we've been having to try to understand the waning issue.
That point about the waning issues, particularly the fact that it was acting as an antiviral, even a weak antiviral that either led to escape mutations or just waning of the antibody concentration, is an important point to remember. Does anybody else want to make any comments on this point before we move on to another question?
Just that Peter's last point, which we're trying to understand, we have seen with antivirals, as you know, with cabotegravir, we've seen that phenomenon, and we have to really search hard to make sure we didn't that there aren't masked infections. Where seroconversion cannot occur, infection is present in the tissue but not detected, except with the most sensitive of assays at some period of time. So that phenomenon almost certainly exists. So
that's a very important point. Before we get to the trial design issues, I have one other question from the biology perspective. Because this study was done in a population of men and in a separate population of women, who were combining the data, what does it say that the data was so consistent about the level of activity in men and women about the biology of HIV acquisition. Does it say something along the line? This is maybe a little bit of a radical statement, that there really are differences in terms of susceptibility between men and women, but the antibody just, it's more about the virus the individual is exposed to rather than the given biology. Is that a valid interpretation of these results, or am I overreaching? And this is to get some sort of debate going here about whether gender or sex matters here, but is it just the virus?
Well, I think we certainly thought that there would be a difference. And there was a lot of in vitro stuff with ex vivo infection that, maybe the vagina was protected better than the rectum. And we've seen these differential effects at times of the antiretrovirals. So the fact that it was the sort of innate resistance of the circulating strain that made a difference here as we relate to antibody was I think, I'll say to myself, it was surprising to me that the cutoff was exactly the same, and that the data in men and women were identical. And I think these kinds of studies really outline to some extent how sort of little we know about the first 24, 48, 72 hours of infection in HIV after all this work that we've done. And what are the real predictive issues? And, maybe Michelle, your thought leader here,
Nobody
throw it back to you as to, what you think about this.
No, I don't really know, Larry, and it's really not my area.
Okay. All right.
It is something to think about. And it's interesting, Mike, that you raised the issue of cabotegravir. Do we see the same sort of general pattern in terms of concentration or drug concentration, same curves with CAB?
Yeah.
Or is that apples and oranges here?
No, it's a little bit. Well, first of all, I agree with Larry. I think Larry and I were both really surprised at that result. That's the first point. Second, I want to ask Lynn a question, or John and Lynn, about the signature sequences. We spent so much time in SHIV about the single founder virus, and I think that Carolyn gave a talk earlier this week in talking about like signature sequences of founder viruses, which is kind of a corollary of this biology. And I don't think we have all the data yet in that regard. Third, with cabotegravir, I can say the following: that the chance of kind of a masked hidden resistance issue was really considerably greater in men than it was in women. So there was a difference. There were more men who had, like, I think it was 15 men that raised the issue of not easily detectable infection compared to only about 4 women. So it wasn't exactly the same. And so the biology isn't exactly the same. Again, meaning that the antibody studies were a pretty big surprise, I think. I was very surprised, just like Larry said. I remember this conversation. But Lynn, can you comment on the signature sequence idea, on the PacBio and all that before we go on?
You're muted, Lynn.
Or maybe Peter.
What Carolyn presented was, the number of multivariant infections in the trial, and in fact there was a higher percentage of multivariant infections in AMP compared to previous studies, but that was actually because they used PacBio, and so they just had greater depth, and so they just found more variants in the trial. So
women and men were the same.
They had 70%, were single variant transmissions, and 30% were multiple.
Were women and men the same in terms of multivariant, Lynn?
So it's just it's more than one. There's minor changes in the. It's not a single virus.
But were women and men the same in the results?
Oh, sorry. Women and men the same. Yes, I think they were. Yeah.
Okay, thanks.
Okay, that's really interesting. So if it's okay with the panel, I think one of the areas that there's been a lot of discussion in the chat has been future trial design. We have now seen the success of the cabotegravir trials, which were designed to be either non-inferiority or superiority of an oral medication versus an injectable. But what is the future of an antibody trial? What would that look like? What would be the control? What would we want to set as either the target product profile, or what would be the criteria that we would need to see from either pharmacology or biologic activity in small human studies before we wanted to head off on an efficacy question? What would that look like? There was one comment in the chat that thought we should also put cost into the equation, in addition to what Larry had put forward as his target product profile. So let's spend some time talking about that. Who wants to go first?
I want to disagree with Larry. I want to start just so I can start a fight with Larry about this. I think that realistically, a couple of things, and this could start a fight with Michelle too, so that'll make me happy. I think that these longer acting agents are really critical. Larry used the word ultra long, and I think that's true. There's got to be an advantage over the pharmaceutical small molecules that have been developed. I worry a little bit about so much focus on sub-Q or even IM instead of IV. We learned that people don't mind IV, and if it's given every 6 months, and if potency becomes this magical thing where it's got to be the world's most potent antibody, maybe that's a mistake, because we can give this stuff IV every 6 months and we know people will tolerate it. And we think in the COVID space, we'll probably end up giving infusions. I mean, as we search for COVID monoclonals that are sub-Q, we may end up in these little CVS doc in the boxes. No one may have seen it, these little huts of giving treatment with IV infusions. So I'm not so opposed to IV at the expense of searching for potency. I think the tissue concentration issue is really, really important, that it's both how long can we make it last, and will we achieve a tissue concentration in the rectum and the vagina that whether we use Michelle's assay, monoclonal assay, or TCMBL, that we really need to feel pretty secure that what we're giving is going to last 6 months, provide tissue protection, and give us like something comparable to a small molecule. I'll stop, and I hope that made Larry.
No fight whatsoever. I put it on the slide for discussion, so it did its job. But Peter sent me an email. This is like a prescient question about the future trial design last night. So Peter, take this on.
Okay. Well, first to say briefly about, thinking about what we'd need to see to go forward with an antibody regimen to an FC trial. And, if we take a serum neutralization titer as being a key parameter in predicting efficacy, then basically what we're looking for is that for a future FC trial, we have to look at the population that'll be studied and understand obviously the schedule of the antibody regimen, and understanding the PK profile across the period of follow-up of the study. And then we have to understand the circulating viruses and that'll be exposing those individuals, and then understand the distribution of resistance levels to those viruses. And then we can basically calculate the percentage of person years at risk that these monoclonal antibody regimen recipients have over the entire follow-up period, that are with serum titers that are basically high enough that, based on the AMP study and other studies, that we can infer that they were probably protected. And so then you can basically predict the prevention efficacy being at least 90%, say, and that would make us feel like it's ready to go. And then to talk about potential trial design, Linda Gale, I saw you pose this question too, and I know the Discover trial is perhaps interesting as, I don't know if a model, but it stimulates some thoughts. That trial was looking at different oral prep agents in a trial with just 2 active arms, and there wasn't a placebo arm. And my interpretation of the trial is successful because the trial showed very low incidence in both of those arms, And then it was credible to do a counterfactual placebo to infer that it was quite likely that there would have been Fs had them in a placebo. So I think the same thinking over in the monoclonal antibody world. I think one option would be to do a trial of say a double mAb or a bispecific versus a triple mAb or a trispecific, and power it for something like 70% efficacy of the double versus a counterfactual placebo and 90% efficacy of the triple versus a counterfactual placebo. It would actually be powered to look at the head-to-head comparison of the double versus the triple. And it might need about a sample size of 12,000 if we assume a background incidence of about 1.5%, and compare, if a trial like that could kind of verify that the triple had higher efficacy than the double, combined with all the marker data that would be collected, same type of data from AMP and additional data, I suppose. I think all those data together could lead for a case for approving an antibody regimen, even though the design doesn't have a placebo arm.
Yeah,
and
Peter, one thing for Peter though, I think that the ethical issue that'll surface is we have to be secure enough about the antibodies, whether it's Michelle's combination or John's. We have to be secure enough of the antibodies that we're not using Truvada in the background. I think that, so this is the issue. If we're going to go like Discover had TDF-FTC versus TAF-FTC, great. They were so confident that there was no background. But in AMP, we haven't talked about it all, but we had to analyze the background contribution of Truvada. That's not going to be acceptable in the next trial. So therefore, the study subjects have to be assured that there's a pretty good chance that the cocktail we choose based on whatever metric we use is going to provide them pretty good protection. I just don't see another design. But again, I'm willing to, and one last thing for Michelle, because and that is, I would argue, Michelle, again, that we've always said in our 1,000 conversations, we could give your antibodies IV. I've now, just to reiterate that point, I'll stop.
So I have a question for Peter about the notion of the study. How many infusions would you want to be able to run a trial for? What was to be the period of time? Let's make an assumption that the two arms had equivalent PK and that they would last, say, let's just pick an easy time of, say, 6 months. How many years would you want to run the trial? And would you then also have to worry about the tail on the antibody, just like we worry about the tail on antiretrovirus?
Yeah, that's an interesting question. And there's always a trade-off with how long you run the trial, where the longer you run it, you gain power by capturing more events that way, versus starting with a larger sample size to get to more events more quickly based on that basis. Yeah, so I think one thing we learned from COVID is that if you start big with the sample size, you're going to get answers more quickly. So I think that's kind of appealing. So I would think that we'd want to use a schedule that is as sparing as possible while still getting those PK curves and the predicted serum neutralization titers high enough that we can meet our benchmark for predicted overall prevention efficacy.
I would chime in. I think the lesson learned from COVID is just what Peter said, which, they designed the trial 15 and we doubled it because we needed speed. And then in the end, when you have that answer and then you have this large cohort, you really can get some really amazing data over time. And I think we as a field need to reflect about that. Yes, that pushes more tension on you, Carl, because larger numbers do need more money. But I think momentum is really important in these kinds of trials.
I just want to say one thing about the small molecule drugs and antibodies. I think the small molecule drugs are amazing. Obviously, they're incredible, and they by themselves are going to be revolutionary should they be taken up by the population. And should there not be a problem with the emergence of resistance to single drugs, which is something that I think everybody worries about. And one could make the argument that because the VRC01 AMP trial, the results, in fact, there was very little protection, that one shouldn't even bother with the other antibodies, really, because they may not be the 100% that the drugs are. But it's really hard to predict going forward uptake of these drugs and whether or not there are going to be additional problems with small molecule drugs. And the new antibodies, whether they are the triple combination, the double, or any of these other things, are LS antibodies, which is really a difference. So I think the question for the field is, do we test any of them? Should we test any of them as agents at this point or not? And I think that's something that we all have to struggle with.
Thanks, Michelle. We have about 5 minutes left. One question came in that is an important question to maybe for people to ponder is what is the value of moving antibodies into, say, vertical transmission? This has been a lightning rod for a number of years. Is it time to consider that, or do we still feel that we will need to get some efficacy data in adults before we head down the maternal and child transmission pathway? I see that is a question that really nobody really wants to tackle. I'll make the statement that I think before we go and do a study in maternal child transmission, we really need to make sure we're doing the best job we can to deliver antiretroviral therapy to pregnant and breastfeeding women. I think that is what we owe them.
Yeah, I mean, I think there's something really attractive about antibodies, long-acting antibodies in breastfeeding, reducing breastfeeding transmission. I think there's, quote, the natural substance, the safety profile as it relates to this, and during this period of time, I think it's just a perceptual issue here that and would be embraced a lot. So I actually think there is a place there, and maybe an excellent place because of the sense of, quote, natural substances. And the trials are complicated, but in the end, I think one should have a very thoughtful discussion about using it during this period of time.
All right, we have 3 minutes left. Does everybody want to take 20 seconds
John,
to sum up?
John wants to talk because I think they're doing a trial. A VRC, isn't VRC with NII doing a trial already in vertical space?
There are, the Impact Network has done some trials that demonstrate safety in PK, so the potential to use antibodies in infants. So, that's a start. I think the adult and maternal or infant questions are interrelated. Going back to Michelle's comments, we have to figure out where antibodies fit in the paradigm of prevention transmission. And, I do think that if you kind of bring us back a decade or so, it was never obvious to me, for example, that small molecule drugs would work as well as they did, right? There were raging debates that it would be impossible to prevent, quotes, to have sterilizing immunity. Those of you old enough to remember those. But in fact, you do get sterilizing immunity beautifully with small molecule drugs. And, and then, will they be used? And there's some toxicities. And now we know antibodies have a pretty high bar for resistance, but they can work. So to me, we don't have a perfect prevention toolbox. And it does seem, I think you need the antiretroviral experts in the mix to design a next antibody trial because they're there and they work. But I do agree with the concept that you could get synergies from approaches that have completely different mechanisms of action.
Thanks, John. We have about a minute left. Does somebody want to make one final statement as we wrap up?
Hi, yes, this is Shri. I saw a question in the chat about how did the participants feel about receiving a placebo instead of PrEP. I just want to clarify that all participants had access to Truvada PrEP in the men trial as well as the women trial. So it was both treatment and placebo arms had access to Truvada. So it wasn't a replacement for placebo.
Thank you for that clarifying comment. That's extremely important, not to leave people thinking that PrEP was not available during these studies. That's an incredibly important point. I want to close by thanking our speakers and then our additional panelists for a very interesting and informative session. This is one of the last parts of the meeting, and this is a great way to go out with a lot of questions and a lot of work that we have to do for this field. So thank you all, and have a good rest of your day. Bye.